Hello, my lovely anatomists and physiologists. Michele Glass here, and we are still talking about transport across the membrane. And here, what we're talking about is osmosis. And osmosis can be described as the diffusion of water. Remember when we did our stoplight? And we said that some molecules get the green light, the go ahead, and those are going to be your lipids and your lipid solubles and your really small stuff. And we said some molecules get the red light, and the red light is your really big, really polar charged molecules. And then other molecules get the yellow light. And the yellow light we said was water. And the reason for this is because, well, water's polar. It doesn't really want to go through the phospholipid bilayer. So I'm going to draw my quick draw of my phospholipid bilayer with my heads oriented on the outside and my phospholipid fatty acid tails on the inside. And what we see is that water is polar. It does not want to go through, but it's tiny. And so it can go through. And so what we see about water is that it can squeeze through, or it can use special channels, membrane protein channels in the membrane to go through if the cell needs it to move a little bit faster. But water is not regulated by the membrane. Okay, so water then, the movement of water across the membrane is based on the concentration gradient. Remember, that's the hill that we drew. So what we're saying with osmosis is that water will go down the hill from an area of a high water concentration to an area of low water concentration. And so this super, super duper matters because dependent on the ECF, the extracellular fluid, cells can gain or lose water. And water, we know, is necessary for human life, right? So water is involved in all kinds of chemical reactions. Water is 70% of the human adult body, right? And so we know that like we can only go a few days without having water, and that's because our cells need that water. And so that makes it obvious, like you don't want your cells to dehydrate, shrivel up. But we also don't want our cells to gain water because remember that cell membrane is very loose and there's nothing else as a border for the cell. And so if a cell gains water, it can just rupture, right? And so obviously that is a bad scenario for the body. Okay, just a few reminders. When we talk about a solution, we're talking about a solute plus a solvent, right? And the solvent is the dissolver. And the solute is what I like to call stuff, right? Because it could be electrolytes, it could be nutrients, it could be hormones, it probably could be gases, right? Could be all of these things. And the thing that's important to remember is that water is a universal solvent. And this is important to remember because when we talk about osmosis, we're talking about the movement of water, right? We're talking about movement on water from a high water concentration to a low water concentration, right? So here's the question. How do we know a high water concentration and a low water concentration? Okay, so let's catch another couple of terms. So let's talk about something that is concentrated. So you could think of dish soap, dishwashing liquid is concentrated. You can think about some of our liquid detergents you can get in these really small packages because they're concentrated. You can think about, I don't know if you've ever made like orange juice from the freezer section. You can just get this tube of frozen orange juice and then you add water. Oh, what about condensed Campbell's soup, right? Concentrated. So concentrated means high concentration of solute. So here I'm doing my upwards arrow and I'm putting solute in brackets to indicate concentration. And then it means a very small concentration of water. If you want to, you can think about this in context of the human body as like salty, right? There's a lot of stuff. There's a lot of solute and very little water. If a solution is dilute, it's the opposite, right? So think of like, ooh, this coffee is too dilute, meaning I didn't get the ratio of my coffee and my water, and I would call it watery, right? And so in this case, I have a low solute concentration and I have a high water concentration. So something that's dilute, we can think of as being watery. Something that's concentrated, we can think of as salty. And so paying attention here, what we should see is that something that's dilute has a lot of water in it. Something that's concentrated has very little water in it. And osmosis, remember, is movement of water from a high water concentration to a low water concentration. I'm using all my shorthands there. And so what this means is water would go from a dilute solution to a concentrated solution. So this might help us as we talk. It's going to go from something that's watery to something that's salty. Okay, so let's take a look at some specific terminology. We have this term next, which is called tonicity. And tonicity is a comparison term. It's talking about the solute concentration and the ECF compared to the solute concentration and the ICF. So two things to pay attention to here. Number one, tonicity is defining the solute concentration. Solute is the stuff. Osmosis is water. Tonicity is defining where is it salty? Where's the most stuff, right? And then the other thing to pay attention to is tonicity is defining the extracellular fluid as it relates, as it compares to solute, to the intracellular fluid, right? So it's a comparison term, it's a relational term, right? And so you have three types of tonicities, and I like to always write mine hypertonic at the top, isotonic in the middle, and hypotonic at the bottom. Iso prefix means same. So isotonic solution means the solute and the ECF, solute concentrations, I put it in brackets, and the ECF is equal to solute concentration and the ICF. There is no hill, y'all. It is a flat land, right? There's no gradient. There's no gradient. So here, what we see in the cell is you have equal movement of water in and out of the cell. The cell does not gain or lose water. So this is like, how do I want to say this? Yeah, I'm going to call this homeostatic normal. No, I'm going to call it ideal. Ideal for human cells. This is ideal for human cells. This is all of the balance of the kidneys to balance your fluids and your electrolytes is all to maintain your extracellular fluid to be isotonic to your cells, such that your cells are not gaining water or losing water. They're staying the proper size. They're being shaped in the way that they work best, right? Okay, hyper is up above because the prefix hyper means above, right? So hypertonic, remember, is going to be a high solute concentration in the ECF compared to ICF. Hypertonic is a high solute concentration in the extracellular fluid compared to the intracellular fluid. If you want to, you can think about this as being salty. In this case, your cell has very few solutes. I'm putting through a circular cell, and then I have a bunch of dots on the outside of that representing my solute. And I have just like 3 dots on the inside representing the solute inside the cell. And so what we're going to see happening here is water will leave the cell. And so basically the cell dehydrates. The cell loses water. It can shrivel up, can look like a raisin, right? Or something else that's like crusty and dry. It's not sustainable for a cell, right? So if a cell loses too much water, you know what's going to happen. That cell's going to be dead. It's not going to be functioning the way that it should be. Okay. And so what has to happen instead here is you have to balance out. You're going to feel thirsty. You're going to take on a lot of water. You're going to retain that water in your body fluids to help balance out all of that saltiness, right? Hypotonic I have listed below because that prefix hyper means below. So hypotonic is a low solute concentration in the ECF compared, of course, to the ICF. So here you can think about a hypotonic solution as an extracellular fluid that's like watery. There's not enough solute. So here I'm going to draw my circular cell, and I'm going to put like a whole bunch of dots inside the cell and just like a few on the outside. And so what we see will happen is the water will go into the cell. And so the cell can gain, and it can burst or lyse. And the term lyse means to burst or break open. And it's not going to break open every time. It can gain a certain amount of water without breaking apart. But ultimately, if it keeps gaining water, it can break apart. And just like you would have a problem if like you ruptured, right? If your cells are all rupturing, clearly that's bad news, right? That's bad news for the body. Your terms tonicity, again, are comparison terms, and they're always describing solute in the extracellular fluid, and it always implies then that you have a cell. So basically, like embedded in the term tonicity, we get this conversation about, oh, what's going to happen to a cell. So that's the significance of these terms. The last thing to talk about here is filtration. Now, filtration is not a type of diffusion. It's something completely different. In filtration, you have a hydrostatic pressure gradient, meaning you have one part with a high pressure and then another part with a low pressure. And it basically pushes fluid basically pushes fluid from a high pressure to a low pressure. Now, here's the thing. Whenever we're talking anatomy and physiology, whenever we're talking about the body and we talk about hydrostatic pressure gradient, I think about the heart. So what we're really talking about is as your heart pumps and pushes the blood through the arterial system is creating pressure. And so what that means is as the blood and all the dissolved nutrients are being pushed through the blood vessels, there's a big, huge buildup of pressure until you get to the capillary, and the capillary is the vessel where material can leave. That's where things can enter and exit the bloodstream. And so finally, it's like this big push, big push, big push, big push, and then you get to a point where stuff can exit. And so at the capillary, you get this filtration of stuff leaving the blood, flowing across the tissue. Ultimately, we'll see a lot of that material comes back into the bloodstream. But this is what we're talking about with filtration. So this is an idea that will come up against pretty heavily when we study cardiovascular system and urinary system in the future. All right, up next, active transport. Stay tuned. And as always, take care of yourselves and each other.